Good morning and welcome everyone. Thank you so much for joining the American Cancer Society for our virtual research lab tour with Dr. Stuart Martin of University of Maryland. Many of you may know that the American Cancer Society is actually the largest funder of cancer research outside of the federal government. And right now we have invested more than $382 million in cancer research around the country, and a little more than $14.5 million right here in the greater DC, Baltimore area. So Dr. Martin has been a longtime supporter and cancer researcher with the American Cancer Society. His lab has been funded for more than 10 years. And from the years 2011 to 2019, Dr. Martin served as the lead for the University of Maryland's IRG. And then for the last 4 years, Dr. Martin has served on the Greater DC, Baltimore area board and is currently serving as the vice chair. Today, he is going to talk to us about his exciting work around cancer metastasis. I'm not even going to begin to describe what he does because he does that so well. So, Dr. Martin, without further ado, please take us on a tour of your lab and your work.
Okay. Good morning, everyone, and thanks to Swana for the introduction and for the invitation to do this virtual lab tour. I'm going to go ahead and try and share my screen, and do bear with me if there's some technical issues. Swana and I have sort of worked through switching to the phone to give a tour of the lab and those sorts of things. But here, if I share my content, I'm hoping everybody can see that there. And so this will be a tour of the University of Maryland Medical School and the Greenbaum Comprehensive Cancer Center. So if you're driving downtown Baltimore, the building that I work in here, where my lab is the tallest building in the area that says University of Maryland here across the top. And I sort of gave you a view here of the downtown area because when we did this tour in the past, we of course did it in person so that we could actually walk through the hospital that's right here. And so our research building connects directly into the hospital. My kids always enjoyed visiting here because our building is a little bit taller than the Shock Trauma Center. So my kids could actually watch the helicopters land on the hospital and bring people in and out. And so the research building here, as I mentioned, is connected directly to the hospital. The hospital is actually due for a major renovation that begins this year, that was funded by a very large donation from the Stoller family and will be the Stoller Center for Advanced Medicine. And so this construction on this begins in 2021. And again, here on the other side of the hospital, we have a direct connection into the research building so that we can communicate directly with the clinical side with cancer patients and move directly into the labs for further research. And we'll talk more also about the Hope Lodge today. And the Hope Lodge is only about a block over to the right from these buildings here. So the Hope Lodge is very close to the hospital and to the research buildings. And so just to move into what we research in my lab, Swana had mentioned that we study metastasis. One of the great challenges we have in treating cancer in patients is the detection limit. By the time a tumor is first seen on an MRI or with a mammogram for breast cancer, that tumor has reached a size of more than 100 million tumor cells. So we're really only seeing cancer at these very large stages. And we've developed a number of drugs in the cancer research field that are good at reducing tumor growth, but not nearly as many that are good at stopping how cancer spreads through the body, which of course is the greatest threat to the life of patients. And so the discovery that we made in my lab was that if we put breast tumor cells into an environment in which they were free floating, as if they were traveling through the blood to distant tissues, what we noticed was these cells formed these protrusions on the surface of the cell that sort of reached out around the cell. And we initially thought these were structures that had been reported before that are based on a protein called actin. And we did an experiment where we took the cells when they were free floating, when they'd formed these small protrusions, and we treated with a drug that destroys actin, that was known to destroy these structures on tumor cells called filipodium and vetopodia. And we made the prediction that if these were the same types of structures, they would be destroyed by this drug. So that was our prediction. We did the experiment, and in fact, those protrusions were not destroyed. They got longer and more numerous. So this was a surprise to us, and it showed us that we were dealing with a structure that hadn't been reported before. We followed up by looking at which proteins were in these protrusions, and it turns out to be this type of protein called a microtubule. And the actin that was found in other types of cells was only really found at these ends. So this showed that we were dealing with a structure that hadn't been reported before. We had the opportunity to name them, and so we named them microtentacles. The reason we name them microtentacles is they play a role in how tumor cells stick to each other. And so if we use a very advanced microscope that I'll show you on the lab tour in just a second, to image 2 breast tumor cells that are free floating, we use a special label to label the cell on the left with green, and then we can see what that cell is doing to the cell next to it. And what we can see is that it's forming these protrusions, the microtentacles, that wrap around the adjacent cell and form these clusters. The reason this has become very interesting in the field of cancer research and treatment is clusters of tumor cells that are traveling through the blood have been shown to have up to 50 times higher metastatic potential. For reasons that are not entirely understood yet. The other thing that we were able to do here is if we labeled a tumor cell with green and allowed it to attach to red labeled cells that make up the blood vessel wall, again with the advanced microscope, we can turn this around and see where the tumor cell is penetrating through the blood vessel wall, but also cut through in cross section. And so what we found were the tentacles are actually able to penetrate the junctions between the cells in the blood vessel wall to allow the tumor cells to reattach. And so we've identified drugs that are able to inhibit tentacles. This is one, it's a local anesthetic, tetracaine. And so this is about a 10-minute drug effect here. But you can see the tentacles are moving very rapidly. As the drug takes effect, they collapse to the center of the cell. So we have a number of different therapies that destroy tentacles. The other thing that we've learned here is because the tentacles are based on these microtubules, there are many existing cancer drugs that target microtubules to reduce tumor cell growth, but they target microtubules by stabilizing them. So if you take a tumor cell that has very few tentacles and give it one of these drugs that's used in the clinic right now, like Taxol, the tumor cells get more tentacles and reattach faster. So this is raising the prospect that some of the drugs that we have that reduce tumor growth might actually make the metastasis of circulating tumor cells worse. And so this is something that we really need to focus on going forward in terms of improving therapies to reduce metastasis. And so then just quickly, we worked with a lab in the bioengineering department at University of Maryland College Park to develop a technology called the Tether Chip, which uses an engineered surface to actually hold the cells in their free-floating environment as if they're traveling through the blood, but still allow us to image them and measure these important phenotypes for whether the tumor cell can metastasize. And so the benefits of this Tether Chip is it's very optically clear, and we can test drug responses in the tumor cells in less than an hour. And so looking at this now, if we hold a cell on the Tether Chip and watch its tentacles move over time, what you can see is in the background, the fluid around the cell is constantly being washed out, but the cell is held in place. Now we can wash in a drug here that destroys tentacles, or a drug like Taxol that actually increases tentacles. And then together, again, with collaborators at University of Maryland College Park, we wrote a computer program that actually counts these tentacles automatically. As much as I tried over many years, I couldn't get students to count the number of tentacles on the cell. But now these computer programs do it automatically. The other major innovation that we just recently reported on the Tether Chip was we engineered the Tether Chip surface to actually capture the tentacles. And so this was important because in the free-floating cells, if you try and preserve the cells with a fixation like formaldehyde, tentacles are destroyed very easily because they're very small. And so a lot of previous studies have been using these chemical fixatives and destroying the tentacles. So a lot of people have been missing observing them. And so what you see here is a cell that's where the tentacles are not tethered. When the formaldehyde comes in, you'll see a brief flash, and then you'll see what happens to the tentacles. They very rapidly get displaced and almost immediately begin to collapse. When we allow the tentacles to tether to the tether chip, now when we put in the formaldehyde, you may, if you look very carefully, see a small bend in one of the tentacles, but most of them stay in place. And so that's here. You'll see a brief flash with the formaldehyde, and then the tentacle bends a little, but it's not destroyed. So this allows us to preserve them. And what we were also able to show here by developing this technology is that tentacles are common in many different tumor types. And we've now seen them in breast cancer, colon cancer, lung cancer, prostate cancer, and we have collaborators here at University of Maryland that are working on them in ovarian cancer. So the microtentacles seem to be a very common strategy for tumor cells to try and reattach. And then I'll show you in a minute when we walk through the lab that we have several instruments for isolating circulating tumor cells from patient blood samples. Basically, tumor cells are much larger than blood cells. So each of these instruments use a different strategy to pull out the larger tumor cells. The angle parsortix here uses a series of sort of stair steps that trap the larger tumor cells at the top. And so this is actually a movie of blood flowing through the parsortix cassette. And what you'll see is the small red blood cells go over the tops of the stairs, but the large tumor cells get trapped at the top of the stairs. And so this is what that looks like. So the blood flows through very quickly. The tumor cells get trapped at the top. And because there's this very large slot for the blood cells, the cartridge doesn't get clogged. The cells flow through very freely. So now we trap the tumor cells from the blood in this cassette. We can image them right away to see tentacles. So this is a cell here trapped at the top of the stairs. We can see the tentacles. The other technology we use is Vortex, from Vortex Biosciences. It basically uses this sort of microfluidic approach to set up these little whirlpools that trap the large cells. And then we were very excited about this because we can place our tether chip immediately downstream of this isolation and capture tentacles very well, preserving them nicely on the tether chip. You can also see the difference in size between a red blood cell here, there are a couple red blood cells in this image, and the tumor cell is much larger and generating these long tentacles. And so anyway, the counting of tentacles that we were able to do with College Park has allowed us to test FDA-approved drug therapies, like this is a drug vinorelbine. And in 1 hour, we can see the differences in microtentacle number per cell and average microtentacle length. And the strong differences that we get with this 1 hour drug treatment have shown us through the statistical analysis that we can get 90% power for this drug response with as few as 11 tumor cells. So that means that the few cells that we can get out of the blood of the patients, we can actually test which FDA-approved therapy will work best for that patient to reduce metastatic risk. So that's where we're taking this. And so then just in closing, we have lots of different collaborators across the School of Medicine, as well as Johns Hopkins and University of Maryland College Park. We're supported by a number of different funding agencies from the federal government, the NIH, the Veterans Administration. The American Cancer Society currently supports one of our postdoctoral fellows, and so this is now an image of the group now. So the American Cancer Society is currently supporting a Ph.D. in physics, Eleanor Ori, who's the one who wrote the program that actually counts the tentacles. But then we have a number of other investigators in the lab, sort of a mixture of people who work in physics, tumor cell biology, as well as preclinical models. And so with that, I'll stop and we'll try and switch over to do a virtual tour of the lab. So this is just down the hall to the lab. Here in this room here, we have a number of our microscopes for imaging the cancer cells. And so our first microscope here is one that operates at very high speed. It also has this little chamber on top that you can probably see that regulates the temperature and oxygen content for the cells so that we can study how they respond to changes of environment and also image them in videos for long periods of time. Our second microscope down here is one that's used for imaging tumors in tissue. And so we do things like our zebrafish experiments here, where we look at tumor cells circulating in the body of a zebrafish. And then our other microscope down here at the end is one that uses lasers to actually give us the three-dimensional images that I showed you in some of the videos. And what you can see here on the screen that's being shown here is actually images from the bloodstream of our zebrafish. And so if you look carefully, you can see these red cells rolling along the blood vessel wall. So those are the tumor cells circulating through the blood and actually grabbing onto the blood vessel wall as they circulate. So you see the blood cells moving by quickly, and then the red tumor cells are actually grabbing onto the blood vessel wall with tentacles. So these are the microscopes that we use to look into the tumor cells in great detail to identify what drugs might be most effective against them. And then we'll walk down into the lab. I don't think there are very many people here this early. You guys are getting up much earlier for this type of thing than other people. And so in the lab here, we basically have a laboratory bench here for one of the students or postdocs. And then they have a desk that's next to that for doing their writing or research on the computer. And then over here, we have our tissue culture facility, which is where we grow all the cells under sterile conditions here so that we can grow the human cells to do our studies, either ones that we grow in culture or ones that are isolated from patients. We have a couple more microscopes here, and then of course the incubators that are used to grow the cells. And if we open that up, I think you might be able to see some of the dishes in here where the cells are growing. So these are the ones that are then used to do studies on the microscope, for example, and that's how we grow them. And then I will also walk down here. So this entire open lab is Cancer Center space. And so our lab stretches down here a few bays, but then next to us are the director of the Cancer Center, the director of clinical research, and the director of cancer surgery. And so again, the students and postdocs do their research here at the benches. And then I showed you before that we have these instruments that actually isolate the tumor cells from the blood of patients. And so the two that we talked about in the videos before are the Angle Parsortix, which is here on the right, and then the Vortex Biosciences, which is the one that takes the cells directly out onto our Tether Chip. And so those were both through partnerships with two different companies that developed those technologies. They were interested in the work that we were doing with the microtentacles, and therefore placed these instruments in our laboratory so that we could study the tumor cells from the blood of patients. And then as we walk back here to my office, there are a few pictures here that were donated by previous students of images they were particularly proud of. And that's pretty much it.